The overall goals of this procedure are to show how to maintain cultures of parasitoid wasps on Drosophila hosts and how to dissect lymph gland and fat body. This is accomplished by first culturing wasps on Drosophila larvae. Animals are then dissected to isolate either the larval lymph glands, which involves peeling off the cuticle and removing the fat bodies and other finer structures to isolate the lymph glands. Likewise, the fat bodies themselves can be isolated using a similar procedure. Using immunocytochemistry and fluorescence microscopy, gene expression and cell-specific changes are visualized in the immune tissues after wasp infection.